For decades, the standard textbook explanation for how life develops has been a concept called the modern synthesis, or Neo-Darwinism. The idea is straightforward. Biology changes slowly. Organisms experience random genetic mutations, and natural selection weeds out the bad ones over millions of years of blind trial and error. That strict biological rulebook was built directly on the foundation laid by Charles Darwin. When he published On the Origin of Species in 1859, he made a specific argument for this gradual, step-by-step process. But Darwin also provided a way to test his own logic. He wrote that if anyone could demonstrate a complex biological structure forming without these numerous successive slight modifications, his entire theory would, in his words, absolutely break down. Following that ultimatum, the scientific mainstream spent the next century confident in their position. The consensus was that life simply does not make massive, sudden leaps. Every new trait requires deep, geological time to emerge. If researchers were to actually witness an immediate, complex biological transformation happen in real time, it would violate the core requirement of the established evolutionary timeline. That requirement was tested directly in the 1960s. A biologist named Kwang W. Jeon was conducting research in his laboratory, looking closely at cellular behaviors. His subjects were single-celled amoebas. They were ordinary specimens, floating in a standard petri dish, doing exactly what single-celled organisms usually do. Over the course of a few months, Jeon observed these tiny organisms undergo a physical transformation that bypassed millions of years of gradual adaptation. He witnessed a biological leap occurring in weeks, not eons. The sequence started by accident. The amoebas encountered a strain of microorganisms known as X bacteria. Normally, an amoeba would consume a bacteria like this for food. They ingested the X bacteria, but they could not digest them. The bacteria survived inside the host and quickly multiplied. It was a severe parasitic infection. The invading bacteria tore the amoebas apart from the inside, killing off almost the entire population in the dish. A small fraction of amoebas survived the outbreak. Inside these remaining hosts, the lethal conflict stopped. The amoebas stopped trying to destroy the bacteria, and the bacteria stopped multiplying out of control, settling into a stable biological truce. According to the standard textbook timeline, developing a functional tolerance between a host and a deadly pathogen requires countless generations of random mutations. This equilibrium happened in months, right in front of the researchers. The surviving amoebas didn't slowly stumble upon a solution through trial and error over thousands of years. They mounted a highly coordinated response to an immediate lethal threat. To figure out exactly how stable this relationship was, Jeon introduced antibiotics to the dish. His goal was simple: kill off only the invading X bacteria and leave the host amoebas intact. The antibiotics worked, and the bacteria died. But almost immediately, the host amoebas died right along with them. The two organisms were now a single functional system. Their biological processes were linked so tightly that killing the bacteria caused the host amoeba to die as well. This is endosymbiotic gene transfer. The bacteria transferred their genetic code into the host's nucleus. The amoeba stored the instructions to keep the bacteria alive. In return, it manufactured the necessary proteins and shipped them back. The cells now functioned as a single integrated factory. This exact microscopic merger is how biologists believe complex cells first got their power plants. About 2 billion years ago, an early cell swallowed a bacterium, kept it alive, and integrated its DNA, creating the mitochondria that currently tower our own bodies. Seeing this level of cellular integration happen in a laboratory shows that organisms can fuse into entirely new species rapidly. The biological machinery executes complex, targeted genetic updates when placed under extreme pressure. We have to look back at the standard set by Charles Darwin, his insistence that a sudden, complex biological change would break down his theory of gradual modification. The amoeba experiment represents exactly that kind of rapid, highly coordinated change. Two separate organisms combining their DNA to survive a lethal threat breaks the rule of slow, random evolution. Other major observations support this. Nobel laureate Barbara McClintock proved decades ago that cellular structures can rewrite their own genetic material in response to environmental stress. Researchers like James Shapiro and Denise Noble have built upon these findings to champion a biological framework called the third way of evolution. They argue that living organisms are not waiting around for lucky, random genetic mistakes. Instead, cells possess built-in intelligent mechanisms that actively restructure their own DNA to navigate crises. Yet, a large part of the academic establishment remains hesitant to adopt these concepts. Many institutions still teach the rigid, slow-moving model of Neo-Darwinism as the only valid explanation for biological diversity. When presented with direct observations like rapid gene transfer in an amoeba, the mainstream often categorizes them as curious exceptions rather than the primary mechanisms driving life forward. The events in the petri dish illustrate a biological reality of constant communication and genetic exchange. Cells swap blueprints and execute precise physical adaptations to navigate survival threats. Evolution is real, but the requirement for a slow, million-year lottery is being reconsidered. These lab results suggest that life can innovate and transform with a level of precision that a purely random process struggles to explain.